So thank you for having me. I thought Lena made a lovely statement about our shared ambition to treat patients, and I will say that I really have enjoyed this conference. It's my first time, and I can see many people who have helped us develop Varespladib, which is the primary molecule of my group's interest. So just to be clear, Ophirex is a public benefit company, but I do get paid by Ophirex, and so that's my financial disclosure. Let me just let me advance this. So general outline of this talk, of course, again, nothing happens in isolation, and just I'll briefly discuss the collaborative efforts that have taken place to bring this drug to clinical trials, which have just started. We'll discuss the compatibility of small molecules with antivenom. Of course, one of the questions we wanted to ask as we were developing this was, would these molecules interfere, add to, or as we did not expect, but we've subsequently found was their synergy with antivenom. And then briefly review our first Varespladib clinical trial, and we had the first patient enrolled on August 15th of this month. For me, it's kind of an interesting milestone. We had our first paper on Varespladib accepted on August 15th, 2016. You can see that Anna Maria was the academic editor, and 5 years to the day from that publication, we got a message that the first patient in our double-blind, placebo-controlled clinical trial had been enrolled. And of course, this is very exciting for me, who my ambition was always to see that Varespladib, if it was a good candidate, would get its day in the court of clinical science. And this picture you can see is actually the plate on which we first made the observation that Varespladib appeared to inhibit venoms, and these are the basis for many assays that we do, which are colorimetric. And I'll discuss this. I'll describe this a little more shortly. Just want to point out, in that 5 years, investigators with last names from A to Z have published on these effects, and part of why I feel so gratified to be in this field is that we've actually cited these papers to virtually every regulatory authority as we get inquiries about what the strengths or weaknesses of these molecules might be, and what's the rationale for why we felt we should get permission to advance to clinical trials. And so really a big thank you to all the laboratories that have extended knowledge about this work. So briefly, again, we were interested in the compatibility with antivenom. The first molecule of our focus was Varespladib, which is the PLA2 inhibitor. And as you've heard in Dr. Patel's talk and others, there's a significant interest in metalloproteases, or the combinations of small molecules to abrogate myotoxicity, for example. These are the types of readouts we get. These experiments were performed by Shakti Vaipuri's crew. And yet again, another collaboration that has arisen from the community of snakebite, snake venom and scientists. And in this assay, we were looking for synergy or interference. And as you can see in the left-hand side, we're looking at SPLA2 inhibition assays, and we found synergy between Varespladib and antivenom, but when we combined it with a small molecule inhibitor of metalloproteases, we didn't see that same synergy. Similarly, we found no synergy between the metalloprotease inhibitor Pronomostat with antivenom or Pronomostat with Varespladib. And what you can see in this vertical vertical bar is a single time point, the 60-minute time point, and those data are represented here in a histogram with all the samples replicated in triplicate and timed at 60 minutes. And we can just look here at a basic schematic for a synergistic assay in which you would take various concentrations of a drug, an inhibitor and antivenom, and mix it in varying ratios from ascending and descending ratios of the drugs in comparison to the other, so that you can see if the combinations interfere, add, or act in tandem to have an effect stronger than either one alone. And in this case, we looked at India's big four snakes. These are the 1 hour time points, and you can see at the concentrations we were using, we didn't have spectacularly strong inhibition by either Varespladib in B or antivenom in A. This was premium serum's polyvalent antivenom, but really at any ratio of the two drug of antivenom and Varespladib, you saw a synergistic effect, whether it was Bungarus, whether it was a crate, Russell viper, sawscale viper, or cobra. We saw this effect that we did not see in any other combination. You can speculate that Varespladib, being an active site inhibitor, is working with antivenom that is binding away from the active site, and you have a two hits on the molecule. Whereas when you combine two active site inhibitors, you may not see this, or there's simply no relation. We have not investigated any mechanism, but it's reassuring to see that there's no interference effect. Again, other people have extended these work, as you saw earlier in abstract from Silva Carvalho with respect to micrurus. I saw this abstract, I was quite excited about it, and Dr. Rowan kindly sent me this figure, and this is consistent with the examination for synergy, that when antivenom and Varespladib were combined, they saw a reduction in liver lesions and kidney lesions. And this is nice to see in animal studies, again, extending work that we have not done, nor can we do. Again, we saw no synergy with Varespladib and Pronomostat with respect to metalloprotease inhibition. But the lesson here is, I frantically texted Shakti, could I borrow some of their figures from Dr. Patel's talk, is that even though we see no synergy between Varespladib and a metalloprotease inhibitor in the enzyme assays, their group has demonstrated synergy when combining a metalloprotease inhibitor with Varespladib. And what the lesson here is that the negative results or the non-results you might see in vitro do not necessarily mean that you will not see an effect. You just have to ask a different question. And of course, this is the value of the biological-based assays. It's a little easier to follow when you do see an effect, but these types of assays really show that you cannot rule out important effects that might be seen, especially when they're in biological systems. So I thank you to Dr. Patel and Shakti for allowing me to use these slides at the last moment. So really, next steps from my perspective is to understand, is this a general case that if you use the blocking SPLA2 with antivenom, would you see synergy? We did have a clue from Yulia Soroseva's lab about 5 years ago when we noticed that the combination of Varespladib with Crofab, which is a digested antivenom, also showed synergy against North American venoms. But again, we only know this for Varespladib, so the obvious thing to do would be to try another PLA2 inhibitor. And then, of course, as you could see from other talks, that pronomostat is one metalloprotease inhibitor that could be advanced, and mirimistat is another very nice candidate that could be advanced. And I think the field would benefit from trying to figure out what's the best one to advance, because these projects cost money and a lot of resources and a lot of time. And so I think it's very useful to have a good understanding of what type of metalloprotease inhibitor might be suitable, either as a general inhibitor or regional inhibitor. And of course, there's also DMPS, which is another candidate that has already been approved for the FDA or by regulatory authorities for other indications and could readily be advanced. And so I think that's going to be a valuable step for the field. I'm keeping this a little bit short because I just wanted to show you this other SPLA2 inhibitor that could be examined if you were to ask this question. We have not asked the synergy question yet, but this is a PLA2 inhibitor that was abandoned by AstraZeneca. In our hands, the second peak, if you do a chiral separation, the second peak is just as potent as Varespladib in vitro, but gives very inconsistent results in mouse studies, but could be a very useful research tool. We've had this custom synthesized, and we have the chirally separated molecules, and of course I'm happy to share these to any research lab that is interested in having it, to please write me, and we can share this. Exciting for us, as I mentioned earlier, is that we have just our trial has just been listed. As you can see, it's new on clinicaltrials.gov. This is a phase 2 clinical trial of oral Varespladib with the standard of care, of course. It is double-blind and placebo-controlled. We've been activated in the US, and we hope to enroll first patients in India this fall. It's a phase 2 trial. We're expecting 94 evaluable patients to be enrolled, probably in a 75-25% ratio. Snakebite is relatively rare in the US, and so we think that we'd probably be able to recruit 25 patients, and with the remainder in India. We are not stratifying by snake type. This really speaks to Cecile's work and that of others, but also to the potential strength of an antidote that is safe in the event of a non-venomous bite, but could in varying degrees be beneficial depending on the type of envenoming. And our endpoints are based on syndrome-based outcome measures. And of course, Michael Abiyanis's work is really spectacularly important to the field. And we are using components of the snakebite severity score that we can measure that are stereotypical as part of our outcomes. And of course, this is a discussion I hope to continue with the field and of course with the group in Liverpool. Again, just a really heartfelt thanks to everybody in the field who certainly welcomed me early on and have been great collaborators, the Fry group, Caswell's group, David Williams, WHO, and of course, CHEMA, and to our funders and to our crew at Ophirex, who work absolutely tirelessly to keep this program running and to have gotten us over the start line of our first clinical trial. So I will take questions. For those of you who've been to my talks previously, Oscar usually is on my shoulder, but it's before dawn, so he's still asleep. But he's a huge fan of CHEMA and attended yesterday's late session. So that's it for me. I'll take questions. I think the discussions tend to be illuminating, and so to that end, I hope that this has been an enjoyable talk and interesting. So